At present, the CRISPR-Cas9 gene editing tool can easily change the genome, but it is easy to cause unexpected editing effects such as off-target effects. To better control the editing of genes, scientists have made unremitting efforts in developing different alternative methods. On the 21st of October, 2019, the research team of single-base editing technology and researcher David Liu of the Broad Institute published the latest genetic editing research results in the top academic journal Nature, reporting a new type called prime editing gene editing technology. The technology directly targets point mutations for precise insertion, precise deletion, and various gene editing without causing DNA double-strand breaks, increasing the types of executable genome editing, and effectively reducing off-target effects. In addition, the technology is expected to repair approximately 90% of known human pathogenic gene mutations, providing technical support for the safer use of gene therapy in clinical settings. This article is entitled Search and Replace Genome Editing Without Double-Strand Breaks or Donor DNA. Off-target effects are a major challenge for gene editing. One of the cores of this gene editing technology is a special protein composed of Cas9 protein and reverse transcriptase. Another key is a special GRNA, pegRNA. Not only does pegRNA bind to a specific DNA region of a target gene, but it also comes with a gene editing template. Guided by pegRNA, the Cas9 reverse transcriptase fusion protein can precisely cut a DNA strand and then synthesize the correct sequence of DNA sequences based on the template. Subsequently, the cell's own DNA repair mechanism automatically integrates the new DNA sequence into the genome. Similarly, the same gene editing is performed on another DNA strand, so that both DNA strands can obtain the correct encoding information derived from the pegRNA without breaking the two strands of the DNA. The gene editing is a permanent change. In addition, thanks to the flexibility of the prime editing technology, researchers can achieve any base conversion, i.e. 12 single base transformations, including T2A. Other single base editing systems can only perform 4 base conversions. To validate the gene editing capabilities of prime editing, the researchers performed more than 170 tests in a variety of human and mouse neurons. The results show that prime editing can not only perform single base editing, but also insert or delete specific DNA sequences, insert up to 44 bases, or delete 80 bases, and the risk of off-target editing is lower. It is noteworthy that the research team successfully repaired gene mutations that caused sickle cell anemia and Tay-Sachs disease using the prime editor. David Liu said, "The advantage of prime editing is that there is almost no limit to the sequence of editing. Base changes are only determined by pegRNA. This latest gene editing tool enables specific gene editing and helps researchers to experiment more easily, develop disease models or study the function of specific genes in the room." According to David Liu, prime editing can help researchers solve about 90% of the more than 75,000 disease-related DNA variants listed in the ClinVo public database. This means that prime editing can be used to develop treatments for mutations and disease-causing genes. For the publication of the results of the prime editing research, Science magazine commented that it is a major breakthrough beyond CRISPR. Professor George Church of Harvard University, a pioneer of CRISPR, said, "It is a big step for genetic editing in the right direction." Brittany Adamson, an expert on DNA repair and gene editing at Princeton University, said, "Although it is still too early, the preliminary results of the study look very good." Eric Sontheimer, a molecular biologist at the University of Massachusetts, said, "Prime editing may not be able to perform large DNA insertions or deletions like CRISPR-Cas9, so it is unlikely to completely replace the mature gene editing tools. Because the researchers want to make any changes, they are all encoded on an RNA strand, and the longer the RNA strand is, the more likely it is to be destroyed by cellular enzymes. Different types of gene editing still require different types of genome editing platforms, but prime editing seems to be developed, and so far, other CRISPR alternatives are more precise and versatile. Currently, the David Liu research team has freely exposed this system to academia and non-profit organizations for further testing and improvement. In addition, based on the prime edited gene editing system, David Liu has created a new company, Prime Medicine,